A patient survives a complex abdominal surgery. The medical sutures hold perfectly, but days later, the surrounding biological tissues tear like wet paper. The surgical wound bursts wide open, a failure known as fascial dehiscence. This catastrophic structural collapse shares an underlying mechanism with a seemingly unrelated pediatric emergency. A child develops a minor illness and within hours suffers sudden, violent muscle spasms and acute dystonia. Both crises trace back to the absence of a single biological mortar. That specific mortar is the essential amino acid L-lysine. Despite relying entirely on this molecule to hold our organs, skin, and blood vessels together, the human genome has no blueprint to build it. We lack the enzymatic machinery to construct its core carbon skeleton. Millions of years ago, human ancestors traded self-sufficiency for metabolic efficiency. We outsourced the production of this structural anchor completely. To keep from physically falling apart, we remain entirely dependent on pulling this exact molecule from our diet. The ability of this ingested nutrient to bind our biology together comes down to its precise chemical behavior at a standard physiological pH of 7.4. This molecular model of lysine features a flexible 4-carbon chain ending in a terminal amino group. In human tissues, it stays completely protonated, holding a powerful positive charge. As an electrostatic anchor, the flexible arm reaches across gaps, locking on to negatively charged structures. This magnetic grasping mechanism provides the physical force driving both the integrity of our physical scaffolding and the tight regulation of our genetic code. Deep inside the cellular nucleus, that same magnetic arm functions as a physical on-off switch for human DNA. 2 meters of negatively charged DNA must be packed tightly into every microscopic cell. The body spools this genetic material around histone proteins, which use densely packed positive lysine nodes to grip the DNA backbone. The magnetic attraction clamps it shut, silencing the genes completely. When the cell needs to activate a specific gene, it snaps a tiny chemical packet onto the lysine residue. This immediately neutralizes the positive charge. The magnetic grip releases, and the DNA physically unspools just enough for the transcription machinery to read the sequence. Outside the nucleus, the liver and kidneys extract free lysine to synthesize carnitine. Carnitine operates as the mandatory cellular ferry, physically shuttling long-chain fatty acids across the inner mitochondrial membrane so they can be burned for ATP. Dropping into a severe lysine deficiency halts this synthesis. Fat pools up helplessly outside the cellular furnaces, starving muscles of energy and causing fasting blood sugar levels to drop. But the vast majority of our lysine reserves, roughly 80% of the total body content, are deployed far outside the cell, heavily locked inside our bone matrix, fascia, and global vascular tree. Within these extracellular spaces, lysine molecules undergo targeted oxidation to form permanent pyridinoline crosslinks. This chemical rebar locks adjacent collagen and elastin fibers together, providing the intense elastic snap our arteries need to absorb constant systolic pressure waves. Delivering this rebar into the tissues is a continuous cellular battle. Because of its intense electrical charge, lysine cannot drift passively through cell walls. It must travel through specific transporter doorways, which it has to competitively share with other basic amino acids, particularly arginine. Ingesting massive doses of pure lysine isolates saturates these microscopic revolving doors, creating a traffic jam that starves cells of incoming arginine. Clinicians sometimes leverage this mechanism against the herpes simplex virus, which requires arginine to build its outer shell. Drowning the local environment in lysine forces the virus to grab the wrong structural block, resulting in a defective, non-functional viral capsid. However, overwhelming these cellular doorways causes collateral damage. The sheer volume forces the kidneys to dump vital arginine out through the urine, proving that flooding the system with a single isolated nutrient routinely disrupts the entire biological balance. This metabolic flowchart illustrates the genetic defect known as glutaric aciduria type 1. In a healthy system, excess circulating lysine is safely broken down and cleared by the liver. In GA1, the precise enzyme required to process that molecular exhaust is broken. Instead of safely degrading, the trapped lysine accumulates and breaks down into glutaric acid. This byproduct acts as a highly potent neurotoxin that floods a child's developing brain. These acids selectively target and destroy the basal ganglia. A sudden spike in lysine metabolism, often triggered by a minor childhood illness, initiates acute dystonic crises, resulting in permanent, devastating muscle spasms. Without a functional exit pathway, the exact building block required to maintain cellular structure and bioenergetic stability rapidly transforms into a devastating internal poison. To monitor these tight metabolic margins, clinical laboratories utilize liquid chromatography tandem mass spectrometry. It functions as a high-resolution molecular coin sorter, separating amino acids by their exact mass and electrical charge to quantify plasma concentrations. But the technology remains highly vulnerable to physical mishandling. Red blood cells naturally store high internal concentrations of free amino acids. If a phlebotomist uses excessive vacuum pressure during the blood draw, the sheer mechanical force literally bursts the red cells apart inside the collection tube. The ruptured cells leak their sequestered lysine directly into the plasma fluid. The mass spectrometer accurately measures the sample it receives, resulting in a massive artificial spike in concentration. This mechanical illusion, known as pseudo-hyperlysinemia, creates a dangerous clinical blind spot. A patient's actual circulating levels could be severely depleted, but the fake spike entirely masks the underlying fascial starvation and prevents physicians from seeing the impending structural collapse. Pathologists protect the data by running a strict hemolysis index to detect ruptured cells before processing. They back this up with automated delta checks, which flag sudden lysine shifts of over 50 micromoles per liter, a biological swing that does not occur without massive medical intervention. But impeccable laboratory data only matters if clinicians understand the dietary inputs. Standard cereal grains, including wheat, corn, and rice, are notoriously deficient in lysine. A patient consuming a highly restricted vegan diet heavy in these grains may hit their calculated total protein targets on paper, yet remain silently starving for this specific limiting amino acid. This leads directly to vascular fragility and failed surgical wound healing. Effective clinical stewardship requires tracking the precise molecular inventory, recognizing that a generalized dietary macro cannot replace the specific building blocks keeping the system intact. From securing 2 meters of DNA into a silent, tightly packed coil, to driving the carnitine shuttle that burns our cellular fat, to locking the arterial rebar into place, lysine acts as the indispensable physical anchor for human resilience. And yet, as we rely entirely on the outside world for this nutrient, specific colonies of bacteria living in our gut actively synthesize small amounts of free lysine. Our own microbiome continuously works to offset our genetic shortcomings from the inside out. Maintaining this architecture depends on the raw material and rebar, but the assembly process itself requires a master chemical signal to begin. That specific initiator is the domain of methionine. Our physiological stability is never truly self-contained. It is a fragile, ongoing molecular negotiation with the environment around us.